Protected inside the ovule, the fertilized eggs are now ready to form embryos. The diploid zygote nucleus undergoes 2 mitotic divisions, and the 4 nuclei migrate to the base of the archegonium. Successive divisions result in a 16-celled embryo. The 4 cells at the base of the archegonium are embryo initials. The 4 cells of the adjacent tier are suspensor initials. The suspensor initials elongate, first pushing the embryo initials up against the wall of the archegonium, and then out into the female gametophyte, where breakdown of adjacent tissue creates a corrosion cavity. The 4 initial cells separate, and each begins to form an embryo and additional suspensor cells, which push them further into the female gametophyte. Intense competition between the 4 young embryos for nutrients and space results in only 1 embryo surviving. Embryo systems in adjacent archegonia proceed through the same stages of growth and competition. Soon, however, 1 young embryo dominates and proceeds to develop further, while the remainder abort. The surviving embryo enlarges and develops a shoot, the apical meristem, surrounded by a circle of seed leaves called cotyledons. Below the cotyledons, the embryo forms an elongated cylinder of cells that represents the stem-to-root axis. A protective root cap covers an apical meristem. Nutrients now begin to accumulate in the surrounding female gametophyte. Water is removed and the seed enters a resting or dormancy stage. Seed maturation is also accompanied by the transformation of the integument into a tough protective seed coat. The seed and surface tissues separate from the cone scale, forming a winged seed. Cone opening and seed release usually take place in the autumn of the 2nd year.